All right, thanks everyone for sticking around for our last talk of the day and our second keynote, Dr. Tadashi Fukami, who is coming to us from Stanford University. He did his bachelor's at Waseda University, a master's at the University of Tokyo, before doing his PhD at the University of Tennessee, Knoxville. Then went on to do postdocs at Tokyo and in New Zealand, before becoming an assistant professor at the University of Hawaii at Manoa, and joined Stanford in 2008. He's made major contributions to the field of community assembly, including, or probably most especially, his work on historical contingency, which I'll be talking about today. He's won many grants and awards, including an NSF career grant for his work on community assembly of nectar inhabiting microbes, and has also been recognized for his teaching, including a really interesting award for integrating his research into the undergraduate classroom. And this was the AAAS Science Prize for an undergraduate course that incorporates faculty research. So join me in welcoming Dr. Fukami.
No? Is it on? Okay. I was told that I have 55 minutes. Is that too long for you? So that should mean I should end by 5:35, 5:33 or so, just to have an agreement. So I wanted to, just like everybody else, I wanted to thank for a great invitation. Just like JP said, I've known this symposium for a long time, and I wish that I always dreamed of being part of it, and I get too old. But then you get quickly too old. But then I got old enough to be a participant, so I'm very happy to be here, particularly on this topic about community assembly. I think of myself as an ecologist, although I also, of course, interested in evolution. But as an ecologist, I always think about community assembly as an ecological process. So it was very interesting to hear all the talks about evolutionary processes. And what I want to do today is to start from ecology, but in terms of historical contingency, but then ask questions about how we can incorporate evolution into this ecological community assembly to get a better understanding of what I'm going to call historical contingency. And so we can start with the local habitat, with the can be the real island or some aquatic island. And then just like anybody else, we can think of species pool. And often immigration is considered, like MacArthur, Watson, and many other people, immigration is considered a key when you want to try to understand species diversity and composition on a local community, the islands or lakes or ponds. But more recently, I think there is an increasing recognition that the more detailed aspects of immigration, particularly immigration history, can be important also in understanding species diversity. So I'll give you I'll tell you what I mean by that. Let's say that we have these multiple local islands, and for the sake of argument, let's say that these are at least initially all identical physically, chemically, and these species are coming from the regional species pool for colonization. Initially, what theoretical studies, but also increasingly empirical studies have found is that even if you have the same physical conditions shared by different local habitats, and even if you have the same species pool where species are coming from, just by having variation in immigration history or order of species immigration, you can end up having a very different kind of local communities. And this is what I'm going to call historically contingent assembly. And this happens because of what people call priority effects, where early arriving species can change the local conditions maybe in a subtle way, so that the subsequent colonizers, subset of them are going to be very successful, but other ones are going to be not successful. And it depends on the identity of early arriving species in terms of the successional trajectory. And that's in contrast with more classic idea about what I'm going to call predictive assembly, where species are going to sort themselves because each species has some deterministic properties, characteristics that determines, for example, how competitive they are relative to each other, to other species in the species pool. And if you know the local conditions, then you should be able to predict what kind of species composition you will get. And in that case, in this hypothetical scenario, you should see an identical set of species occupying these multiple local habitats. But as I said, more and more with the data, we are realizing that this priority effect could be more important than we previously thought. And it's not just for, I've been thinking about species diversity and composition, but it's not just composition, but also functional properties of communities and ecosystem could be changed. And I'll just highlight in just a few examples here. For example, there's a paper in Oikos in 2007 showing that if you play with the introduction order of some algal species, then total biomass of that community can be changed, not just the species composition of those algal communities. In the same way, there's a now increasing number of studies on grassland communities and looking showing that the plant total productivity can be affected by historical differences in immigration. And then I had a paper showing experimentally that if you change the order of introduction of wood decay fungi, for example, then how much wood decomposition and the rate of nutrient cycling, those things that we consider as ecosystem properties can also be changed to a great extent just by having this variation in species immigration history. And then on top of that, the increasing realization that it's not just the ecological phenomena, but also evolutionary processes could also be affected. For example, Jonathan Rosas, Luke's advisor, claimed in 1998, although hearing Luke's talk today made me think that maybe this was a little bit overemphasized. But what he was saying here is that even if you have the same species colonizing some different island, depending on which other species came first or late, whether that same species can diversify or not, can depend, or how much they diversify. That can also depend on these order of the different species immigration. And a similar story was told on the plant communities. For example, these plant communities on Micronesian islands, this is indirect. It have to be indirect because you can't really watch evolution happening, except that when you have a bacteria for experimental evolution, showing this in this picture, what we were able to show is that if you played with the introduction order of different genotypes of particular bacteria, you can see we were able to show that the extent of diversification, in situ diversification, can be much affected by immigration history. So what we have is this simple relationship where the history is affecting community structure like diversity and composition. That's what I'm going to go in historical contingency. And as I said, structure can be affected function like a nutrient cycling, and that can be maybe feeding back to each other. And so when you look at it, it's a really disturbing picture. It's a simple picture, but it's a disturbing picture to community ecologists. And that sentiment was summarized in a nice way by John Lawton when he published this very provocative and well-cited paper in 1999, a while ago. But he said community ecology is a mess precisely because of this kind of historical contingency. Simply, we just don't have, in most cases, it's just not possible to gain historical information that's needed to understand community structure if immigration history is really important. So we're kind of stuck if we have history actually being important. And I think, so that was 1999. I was able to, I had an opportunity to review the current literature in community assembly, and you can see that you had the right theme here, because there's, at least according to the number of articles, there's the interest in community assembly is just exploding. But if you look at a little bit more carefully what they are talking about, these papers, they talk about, a lot of them talk about functional traits or phylogenetic diversity or trait-based things. And then I try to be as inclusive as possible in thinking about what papers are thinking about historical effects. So I included priority effects or alternative stable states or multiple equilibria and those things that I could think about. And you can see that this very small fraction of those papers on community assembly are talking about historical contingency. And I think it's because of this fear that if history turns out to be important, what to do? We can't really find a way to advance community ecology. I think that's one of the reasons why historical contingency hasn't been so popular in this literature. So how to make a progress? One way is to change the question. So instead of trying to understand each instance of community assembly and in which case history is important, we can maybe give up that question for a little while and say instead that we want to understand under what conditions, ecological conditions, history may be important, and under what other conditions history may not be that important. So can we get some general ideas about the conditions under which history is important? So that's the question, overarching question, I think can be productive for two reasons. One is that as any science, we are interested in predictability. So to understand under what conditions communities are more predictable and under other conditions predictability is low, that itself is a good understanding, I think. A second reason is that there is an increasing interest, I think, in beta diversity. So not just the species diversity within communities, but among communities, as you know. And this priority effect and historical contingency actually can be an important source of beta diversity. So if we can tell when history is more important and so consequently, when beta diversity is expected to be high, that kind of understanding can also be useful for community assembly if we want to understand species diversity, for example. And so basically, this question is asking, can we have something about ecological context that determines the thickness of this arrow going from history to community structure? And there's an increasing number of studies on that over the past decade or so. And so I'll give you 3 examples just to give you a feel of it, start with my own example. So as a PhD student, I was doing some microcosm experiments with freshwater protozoans and some rotifers. And I found that if you have a small habitat, then priority effects become really important. And when I used relatively large containers to do this assembly experiments, introducing different species sequentially, then I was seeing more predictable assembly. As a second example, there's a very nice paper by John Chase in 2010 showing that when you have experimental ponds with lots of nutrients added, then that's when priority effects are really, really strong. But when you have a nutrient-poor habitat, that's when you have a predictable assembly happening. Just as a third example, this is a more recent paper that I published with Rachel Vanette, who happened to be a participant of the last year's ECSS, and also, as you know, as some of you know, the graduate of this program. She was a PhD student here, and she came to my lab as a postdoc, and then now assistant professor at UC Davis. But in any case, when she was working with me, we did an experiment with this Nectar inhabiting yeast species. And what we found is that if you have a benign, quote-unquote, benign habitats, In our case, it's a low sugar environment. I'll tell you more about these nectar yeasts later. But as opposed to harsh habitat, this is when we did the experiment with the nectar with lots of sugar in it. It's going to be a challenging condition. But in any case, when you have a harsh habitat, it was predictable, as opposed to contingent assembly under benign conditions. And all of these examples seem to suggest that the reason is actually not that complicated. In all of these cases, what happened is that when you have a conditions that allow a really rapid population growth, then that makes species, whenever they come early, they grow up really quickly, and then it will lead to strong priority effects. But when you have a harsh, for example, harsh habitat, then population growth is slow, and so by the time the second or other species come, the conditions, local conditions are not changed so much so that there's not much effect of immigration history. So that was kind of a quick overview of the current state of the studies on historical contingency. And what I wanted to do today is to think about how we can put evolution into this picture. Although historical contingency is actually, rather than in ecology, I think there's a conceptual root in evolutionary biology. I forgot who that was when I think it was also Luke who mentioned Stephen Jay Gould about Wonderful Life. And he had this very nice idea about replaying life and replaying the tape of life. And he was, as Luke was saying, emphasizing historical contingency. So it's interesting to me that within the community assembly perspective, I think this kind of research is a little bit advanced in ecological literature, but not so much in terms of evolutionary biology. In relation to, of course, there's a lot of work, but in terms of these questions, when should history matter? And so I have 4 stories that I wanted to tell, if I have enough time to do all of that. So the first thing I wanted to quickly do is to say that over millions of years, we can still think about not just ecological time, we can still think about how ecological context may adjust the importance of history on community structure. So that's one thing. And I'm going to use a plant example in New Zealand as an illustrated example for that. And as a second thing that I wanted to talk about, I want to say that it's the combination of ecological and what I'm going to call evolutionary context. There's going to be a joint effect of these two things that can mediate the importance of history on community structure. And thirdly, I want to talk about a simulated computer simulation. It's completely theoretical at the moment, but I have a computer simulation study I want to tell you about that show that sometimes when the predictions about how ecological contexts affect the importance of history can be completely changed as soon as you consider evolution, particularly rapid evolution, in the community dynamics. And then lastly, I was slow in realizing how exciting this genomic research is, but now I quickly realize how exciting it is. So I'm going to use Nectar yeast system as an example to say that there may be a way to use genomic approaches to understand all of these in a more precise way, in both ecological and evolutionary context. Okay, so let's start with the first one. And so the question is how these type of predictions about the importance of history, how this could be applied to evolutionary diversification. All of the 3 examples that I just told you all just the ecological processes of community assembly. Can we apply it to much longer evolutionary diversification? So the idea is that, let's say, over millions of years, can we have some niche preemption so that the early arriving lineages on, let's say, an island like Hawaii, or in our case, in my example, it's going to be New Zealand. If there is a plant lineage that came to New Zealand early, maybe there's a lot of open niche spaces so that they diversify and they become abundant. But if you are late arrivals, it's just so crowded, just like maybe progression rule, that they are not diversifying. But what I want to do is to ask additional question to say that do we see stronger niche preemption, which is one of the mechanisms for priority effects, in more benign environments as opposed to harsh environment? And to do it, we had good data in New Zealand. So I had New Zealand collaborators in New Zealand, and Devin Leopold is one of my current PhD students. He worked with me and the people in New Zealand to do analysis focusing on alpine plants in New Zealand. And the genera listed here, 17 genera, are all monophyletic in New Zealand, and there is some estimate, some of them are not very good, but some estimates for each genus in terms of when they arrived in New Zealand. So can we see some patterns in terms of the extent of diversification and also abundance? And can we see something about effect of environmental harshness? So to do it, we combined molecular phylogenetic data with the good field data on abundance of different genera that I showed you here. And what's key here is that in this part of the country, you can see that like in any other place, I think in the world in general, for alpine plants, as you go up the elevation, it becomes just more harsh conditions. I think that's generally well accepted. And so what we wanted to do is to see first whether we see effect of this, the timing of arrival relative to each other. So we have a stem age, and then here we have how many. This is the richness of each genus represented on y-axis, sorry, this is the abundance, relative abundance of each genus in the community, and this is the relative richness of each genera in a community, and each dot represents a genus in some very small local plot. And as you might expect, if you have in all the lineage is more abundant and more species rich. You can see that, although there's a lot of, it's messy, but what we wanted to particularly be interested in looking at is whether we can see some interactive effect of stem age and also elevation. And for the relative abundance at least, what we see is that in the low elevation, which is benign habitat, the slope is steeper than harsh habitat. And so what you can conclude, although it has to be indirect, is that as you go up the elevation and it becomes more and more harsh conditions, then effect of stem age becomes weaker, meaning that the priority effects that might have happened over a very long evolutionary time may become weaker as you go up the elevation. And so, as I said, that's indirect evidence because you can't really do experiments on evolution, particularly if you are dealing with plants. But my collaborators, mostly their work is not really my work, but they tried as hard as possible to make this inference as strong. And so what they did is they used a lot of paleoclimatic data to estimate the free niche space that might have been available when each genus came a long time ago, and then see whether there's any relationship between lineages. And they did find an expected pattern that if you came really early, then on average, they were experiencing more free niche space. And then free niche space resulted in higher species richness for that genus. And in turn, this species richness resulted in a greater niche occupancy by that genus. So it's all indirect, but it's all consistent with what we are expecting. And so maybe also over evolution, not just ecological time, but over evolutionary time, it's possible, it may be possible to say that under harsh conditions, that's when you should expect to see predictable assembly happening. But if you go to a benign habitat, the evolutionary priority effects may be stronger. So that's what I wanted to say about the first part. Then I want to switch gear to think about the joint effect of ecological evolution in context. And by ecological context, I've been thinking about local conditions like nutrients and the size of the habitat or the environmental harshness. But that's really not independent of the species that are coming to these ecological contexts. So where the species in the species pool came from in the first place may be needed to think about, because it's the combination of these two things that can determine how well each species might do. So for example, we can think about how quote-unquote pre-adapted different species are to these new ecological contexts they are colonizing to, and can we use that to have some predictions about when history becomes more important? For that question, the one bacterial species called Pseudomonas fluorescens that I told you about a little bit is really handy, because there is a classic paper in 1998 showing that these are the plant-associated bacteria, but you can isolate them from the field and put this in lab conditions. And it turns out that you need only a few days or several days for you to see rapid evolutionary diversification from a single genotype and to occupy different niches in these lab conditions. So by using this model system, Matt Knopp, one of my former PhD students, did an experiment with me and Samantha Ford at UC Santa Cruz. And what we did is we had ancestral genotype and evolved genotype. I'll tell you what we mean by evolved genotype in a minute. But it's a classic priority effect experiment, just a typical priority effect experiment, I should say. So in one treatment group, we put one of the genotypes and then second ancestral genotype. And the second treatment group was identical. We just flipped the order so that we can look at the order effect. And then we had a monoculture as controls. And to explain to you what I mean by evolved genotypes, we use two types of genotypes. One is a poorly adapted one, and the other one is well-adapted one. Simply, we just allowed these ancestral genotype to grow in these rich media for either a few days or I think a few weeks, and so that they have different amount of time for adaptation into these lab medium. So I'll show you what we found here. And first, when this evolved genotype is poorly adapted, what happened was that whichever genotype you introduced first was very successful in mutation leading to a lot of different morphotypes, which is also genetically based. So there's more evolutionary diversification happening if you are the first one but not the second one, because the second one is suppressed. But when you did the same, so history is important, but when you used the well-adapted genotypes as the dark blue one, what happened was that there was not much difference between, so I'm just now focusing on the dark blue one, the evolved genotypes. When you compare top and bottom, in the lower part, you can see that the amount of diversification as measured by number of morphotypes is not that different. So the obvious thing we want to do is to explain why we get this difference. And I have to say that we have to do actually more work to be completely certain, but there are a lot of data collected not just by us, but this is a model organism, so there are a lot of different labs in the world who are trying to understand the evolution diversification of this bacteria. So based on what we know about this particular bacteria and what they do in these lab conditions, we think we have a really clear, a good idea of what may be happening. And I'll use the same, I don't know if I am referring to Luke a lot now, because I happen to use the same fitness landscape picture here. But I think what can be happening here is that when you have an ancestral genotype, these should be very unfit because we have these lab conditions that are very different from what they used to be living in. They are, as I say, plant-associated, root-associated Pseudomonas genotype. And so suddenly they are thrown into this just uninteresting lab media, and so they're unfit. But as you allow them to grow under these lab conditions, eventually they will start to adapt and trying to climb one adaptive peak. But if they are just poorly adapted, only just a little bit adapted, they still have the capacity to find different adaptive peaks if they are given the chance, so they can diversify. But if you allow these genotypes to adapt and adapt and adapt, eventually they find one peak, and once that happens, population geneticists would tell you, as some of you may be population geneticists, that it's difficult for new mutants to persist because they're not that competitive compared to the ancestral one, and also the finding other adaptive peaks have become more and more difficult. And so these are competitive ones, but they have lost the ability to diversify. And so when you look at this difference, what we think is happening is that if you have a poorly adapted genotype, then they will diversify if they are given the chance. But because they're not so competitive, if there is an early comer, they will be suppressed, so they are not diverse. They won't diversify. On the other hand, if you have a well-adapted genotype, they will be competitive, but because they lost the ability to diversify, regardless of when they come relative to the potential competitor, they will be there, but they're not diversifying. So that's why we don't see a historical effect in this case. Yeah. So it's just one small bacterial experiment, but I think as a concept, I think it's interesting enough to think about whether this type of basic idea could be applied to other systems. Basically, what I wanted to say is that if you have a regional species pool that consists of many well-adapted species, maybe things will be really predictable. Although, on the other hand, if you have a pool consisting of many poorly adapted species, that may be when you have to think about priority effects potentially making a big difference for community assembly. All right. The third thing I want to talk about is rapid evolution. And so I go back to this picture, but those of you who are community ecologists should be unsatisfied with this picture. One big thing that's missing in here is that there is no dispersal among local communities. I've been ignoring that for now. Another thing that may be unrealistic about this picture is that often regional species pool is not independent of local communities. In many cases, it may be that the local communities themselves have to be the source of pool species for other communities. In that case, this set of communities, now meta-community, will have to function as regional species pool. with this maybe sometimes more realistic scenario, when you think about what priority effects may do, I think priority effects could be metaphorically considered a double-edged sword in terms of species diversity in the region. I'll explain what I mean by that. So as I've been saying, I think one possibility is that if you have a priority effects, if you can get a lot of variation depending on immigration history, then that could contribute to better diversity, as I said before. And so variation in species composition among local habitats can happen, and that can enhance species diversity in the region. So that would be a good part of priority effect for regional species diversity in this kind of scenario. But if you don't have a external species pool, what can potentially happen is that because of priority effects, if there are by chance some local communities that happen to be a little bit more abundant in the region, there could be a snowball effect, so that, those communities are going to dominate because of positive frequency dependence at the regional scale. And so over time, there's going to be a regional monopolization of all these local communities. And then because of that, no variation in immigration history, and then priority effects themselves are going to erase priority effects, and then there's going to be a reduction in species diversity. And okay, I put that up too early, but so the question you can ask then is, in relation to evolution, is whether rapid evolution can change this naive thinking. More specifically, if you have a rapid evolution happening in the region, could that erase the bad side of priority effects in terms of regional species diversity? So I already said that it's difficult for priority effects to be maintained if there is no external species pool. And that's not just my vague thinking. There is a really good solid paper, 2004, showing that if you have meta-community, it's very difficult and probably impossible to maintain priority effects in the region. The question I want to ask here is whether if you allow species to evolve rapidly, can that then allow regional maintenance of local priority effects? To ask that question, it's a complicated, to us, a little bit complicated question to start with an empirical test. And so I had a good postdoc, Maike Wittmann, now in Vienna, doing a second postdoc. But she and I worked on a simple computer simulation. If you're interested, we have that available on bioRxiv. And it's as a simplest case, we just allowed, we tried more than 2 species, but as a first step, we assumed that there is only 2 species in this meta-community, and there are many local habitats, and some local habitats are occasionally disturbed so that the new round of community assembly can happen. And we didn't have any external species pool because that's the scenario that we wanted to look at. And then we compared 3 different scenarios. One, we had a fixed traits and fixed species, and to have priority effects, we had 2 traits given to each species. One is the general competitive ability. Another one is tolerance to habitat modification by other species. And so the scenario here is that in a local community, if species A comes first, then that species A is going to make the local habitat more favorable to themselves. And we assume that so that we can have a classic inhibitory priority effect. And so both of the species have just just a fixed trait, and to allow local priority effects. Then we allowed both of these traits to evolve relatively rapidly, but independent of each other. Then we thought it would be more realistic to think that there's a trade-off between the two, so that species, individual, can be competitive, but they are not so good at tolerating the bad things that the other species is doing, that would be a source of priority effect. And so or they can be not so competitive, but they are very good at tolerating the habitat modification by other species. And so I'll show you just a little bit of data here. I'm just going to plot regional frequency of species, one of the two species, over time. And all of the simulations started with the equal abundance. And each line here represents replicated simulation. And so what we see here is that, for example, this green line, what it's showing is that the species 1 eventually dominated that region. And in this case, for example, species 2 dominated the region. In either way, it's not possible to have both species coexisting, and that's consistent with what we've known from Shuin et al. 2004 paper. When you allow species to evolve without trade-off, it's worse actually. The once species become more abundant, they just evolve to be more competitive, and then that's the end of story. So the species only one species can survive there. When you have a trade-off in evolution, different things happen. In this case, what happened was that species coexist by evolving to avoid local priority effects. So the common species is there, and they're very competitive, but they're not good at tolerating the other species. But that's OK, because the other species is so rare that it doesn't pay to be tolerant of other species. On the other hand, the rare species are always experiencing the other species, so they are more tolerant, but they're not that competitive. You can also have different conditions, and in some cases, species coexist despite continued operation of local priority effects. I'm not showing all the data to convince you, but even though the priority effects continue to be strong, sometimes species were able to coexist. And so, yes, I think just theoretically at least, rapid evolution can actually erase that part of the knife, sword, so that if there is a trade-off in trait evolution. And so maybe one possibility that we can think about is that even though it's not possible to maintain priority effects if you don't have evolution happening, by having some rapid evolution, it may be possible to have a priority effect. So we can think about rapid evolution as a way to understand when to expect history to be important. And so that's the 3 things I wanted to say. How much more time do I have? 50, great. And so I think hopefully it's more interesting now. Lastly, I want to talk about genome, what I'm going to call genome-based approach. So for that, I'm going to go back to the Nectar. I'll tell you more about Nectar microbes and what Rosie said about having a model 1, having one taxonomic group in one region. That's been the key to be a good ecologist or good biologist. I always had that in mind, but I was too patient about it. And so I kept switching my study organisms. But I think finally I found my favorite system, which is the Nectar microbes that I started to use after I moved to Stanford in 2008. And so since 2009 or so, I've been using these bacteria and yeast that live in flowers, particularly in nectar, as a model system to think about community assembly. So I want to tell you a little bit about what I've been doing and then try to connect it to what I'm thinking about in terms of genome-based approach. So earlier at Stanford, I had a really good postdoc, Kabir Pei, who now became assistant professor, my colleague at Stanford. We liked him so much that he stayed. And then Melinda was a PhD student. And so with 2 of them, we started to look at what we can see in this nectar. And what we found is that there are multiple species of yeast that you can find in nectar, and also, as I said, also bacteria. And we think this is a good system for community assembly research for 3 reasons. One is that, like any microbial system, we have a rapid growth, short generation time. So if you want to do some experiments, to do community assembly experiment, microbes are always a convenient system. But unlike most other, maybe all other microbial system, or most, I should say most other microbial system, it's a simple system. We are not dealing with thousands of species like you might find, for example, in soil. It can be daunting to understand a high diversity of bacteria and fungi and other microbes. But in this case, because we have high sugar in nectar, it turns out there's only a handful of yeast and bacteria species that can withstand high osmotic pressure. So because of that, we have a relatively tractable system of just a handful of species interacting with each other in a local habitat, which are flowers. And so high sugar. And then, but in contrast, there's really no low nitrogen, which becomes important later in my story. And then it's a well-replicated habitats and well-defined habitats. Each flower can be considered separate local communities because many of them, although not all of them, many of these microbes really are specialists in living in nectar. There are almost no other places you can find, no other places you can find those species that we see in nectar. And so when you think about how dispersal happens, it turns out that they rely on pollinators. In our case, we use Mimulus aurantiacus that are native to California, which is pollinated by hummingbirds. When hummingbirds come to a new flower, they're getting nectar. But if they come from old flower to new flower, if old flowers are filled with many species or at least many cells of bacteria or yeast, they can transport those microbes to a new flower. And that seems to be the main way of dispersal for these microbes. And that's convenient because by tracking where hummingbirds go, or in Robin's case, it's a butterfly. But by knowing where pollinators go, we can think about how dispersal may be happening. Another thing you can do is to catch a lot of hummingbirds and see what they are actually carrying on beaks. And so what we do is to just let them drink some sterilized sugar water, but we don't allow them to drink all of it, and just and the remaining water we just analyze to see what microbial communities there are. And what we found is that individual hummingbirds can be very different in terms of what species they are carrying, indicating that there could be a lot of variation in immigration history depending on which flower you look at, because if flowers are visited by different individuals of hummingbirds. And so we look to see whether there's any priority effects to start with, because that has been my main interest. And so I'll show you just a little bit of data here. What I'm showing here is just one of the species of yeast, Candida rancensis, and how abundant they may be in this lab experiment, this simple lab experiment we did. And what you can see here is that if the Candida rancensis was introduced first, then they become really abundant. But if you put another species first, like the experiment that I showed you with the Pseudomonas experiment, depending on which species of other yeast you use, they are completely suppressed. But it depends on the identity of the first species that we use in the experiment. So depending on which species you use, there is a very strong priority effect. And we were able to explain, not beautifully, but to some extent, we were able to use phylogenetic distance of different species of yeast to explain the strengths of priority effect. And going negative here means strong inhibitory priority effect. meaning that if you have a closely related species, there is a strong negative priority effects. But if you have a distantly related species, then priority effects are not strong. It doesn't mean that they are not interacting necessarily. It doesn't necessarily mean that they're interacting only weakly. They can be affecting each other very strongly, but it's deterministic. One is always very competitive than the other if you have a distantly related species. And we did a lot of experiments, and one thing we've been finding is that the key is the amino acids for competition. Another thing I wanted to show you here is that it's not just yeast, but as I said, we have bacteria, and there's also a strong negative inhibitory priority effect between yeast and bacteria. And on top of that, we also have been looking at how each of these affect for example, the visit by pollinators or the seed set by different flowers. And our experimental data suggests that if you have a bacteria, then there is a negative effect for plant pollinator interactions, meaning that the pollination is reduced and seed set reduced, nectar consumption by hummingbirds, and we also did an experiment with honeybees. They're also reduced. On the other hand, in contrast, if you have yeast in flower, then we don't see an effect. There's no effect. That was a little bit surprising to us. But what this tells you is that the priority effects between yeast and bacteria can have functional consequences for plant pollinator mutualism because, for example, if you have a yeast suppressing bacteria because of priority effects, that can indirectly helping plant pollinator mutualism. So I think it's all interesting, but it's kind of a classic ecological study, interesting but old-fashioned. Do I have 5 more? Okay. So I don't think I will be able to finish everything in 5 minutes, but I'll tell you a little bit about what I mean finally about a genome-based approach. So it's old-fashioned, and we wanted to go genomics for 3 reasons. One is, of course, as biologists, genetic mechanisms is one of the ultimate things that you want to have as a biological understanding. Another thing is that if you have genomic information, I think that can lead to a more deeper evolutionary understanding of priority effects because you can look at different species and which traits and which genes that are responsible for different traits that may be important for priority effects. Those things can only be understood by looking at more deeply into the genome of different species. And the third motivation for thinking about genome is that, so unfortunately, we don't have Saccharomyces cerevisiae, the baker's yeast, in our nectar. We don't see them, but they're reasonably closely related. And so we know a lot about what's happening in baker's yeast. So can we use what people have found out about these baker's yeast to think about these nectar yeast? And so I cannot do this genome sequence myself, but luckily at Stanford there are a lot of good postdocs, and Manpreet Dami is one of the current postdoc I have in my lab, and then Thomas Hartwig is a postdoc in another lab, and he has his own thing, but he got interested in thinking about nectar, so he's working more and more with us. They did genome sequence, de novo genome sequencing of the most common species of yeast, Mechanicobius ricoffii, and I'm summarizing their work in 1 slide, but this is a lot of work, but it's very impressive. They know how many chromosomes there are, and then it's a 15 about megabase, and there's a complete mitochondrial genome and 600 genomes, and then 96.4, and that's really good. Just to give you a reference, Mechanicobius bicuspidata, some of you I think work on that species. Even though that's well studied, I have to. I'm proud to say that this number is better than what you have for my bicuspidata, and then and also Saccharomyces cerevisiae is 97% or so. So we have a really complete I think, although it's not published yet, genome, because of all the hard work that Manpreet and Thomas was doing. And then so once we have genome, we can do a lot to understand potential genetic mechanisms. And one of the things that they found is that there are a lot of gene duplications happening in this particular yeast species, and many of them involve some nitrogen metabolism. And as an example, there's this gene called GAP1, which is important for getting a lot of amino acids into cells. And this is a scavenging transporter. And here I'm showing you, so if you can focus on these, this is the Saccharomyces cerevisiae. These are different genes and how they're related in terms of substitutions of sequences. And this is another well-studied fungi. And you can see that for our species, Microbial Rucophyi, there are 4 genes that are duplicated. Originally in Saccharomyces cerevisiae, there's only 1. And so there are 4 gap genes in Rucophyi. And then they did looked at the gene expression of these different different genes, all these genes. And HIP1 is just a control one to show that if you have a nectar-like environment with only a little bit of amino acids, and if you compare that to standard media with lots of amino acids, there is not much difference here. But if you look at all of these gap genes, all of them are much more highly expressed if you have a amino acid challenging environment, meaning that these these genes that are responsible for taking up a lot of amino acids, which is really limiting in nectar, is really important. And so it's just one species, but we are planning to sequence all of these species that we find in nectar so that we can look at the differences in different species. And my dream is that maybe by looking at the comparing genomes, we can look at the genes and then use that to predict the strengths of priority effects. I'll probably should stop here and then skip all of these to get to the overall conclusion. And so I think we can predict when history matters to community assembly. As I said, there's a tendency to try to, I think, ignore contingency. But I think there are some productive way of thinking about historical contingency. And I was trying to say that we can try to integrate ecology and evolution to improve this prediction. Thank you very much.
Okay, so let's take a few more questions for Tad, and then we'll open up the rest of the remaining time also for the other speakers.
Thanks, that was a great talk. I was wondering if you've looked at the relationship of the diversity of the plant community and then the diversity of or interactions in the microbial community in the nectar. So thinking about the fact that different species of plants are going to produce different types of nectar, and this may change the ecological context that these microbes are evolving in, and that a pollinator may go between different plant species. And so have you looked at all?
Yeah, that's so if I understood the question correctly, the plant individual may be different, may vary in terms of how they might interact with the nectar microbes. The only thing that we've done, or Rachel Vanette has, only thing that we've done is what Rachel Vanette did. She went to different plant species, plant individuals of Mimulus aurantiacus to see how receptive they are in microbial growth in nectar by putting different microbes into nectar. And she saw a lot of differences, consistent differences. So it seems like different individual plants are different in how much microbial growth they they allow. But what we don't know is whether that's environmental or genetic. But it's interesting to think about how plants may control how receptive they are to the microbial growth, and how that might affect their interactions with pollinators. Based on
patterns of species abundance on a trade action. So I use models for that. I simulate models. And I use two assembly topics. One is stochastic with integration, and the other is deterministic, where I start with a supersaturated community, and then I let the species work themselves out. I also study two types of niche crosses. One is the Tillman's community colonization trade-off. So where species can be better or worse competitors, and they trade that off with being better or worse colonizers. That's where systems come from. And the other is the classical MacArthur-Levins model, where competition is strictly a function of trade distance between species. I noticed that in the competition colonization trade-off model, the type of pattern I get is completely different, whether I am using the assembly that has integration, or whether I'm using the assembly that starts with all the species there and I wait for them to settle. Whereas in the MacArthur-Levins model, it doesn't matter. I always end up with the same type of pattern, kind of more deterministic. So it's looking like the competition colonization trade-off model has priority effects, whereas the MacArthur-Levins model does not. Do you have any idea why that could be?
I would have expected the other way, that if you have a competition colonization trade-off, priority effects becomes less important, because by definition, I think competition colonization trade-off makes the less competitive species tending to arrive first, and then the strong competitors eventually show up, and then that's it. And so I thought I would have expected under that condition, the communities are going to be the same. So the fact that you are getting the opposite result,
Well, that might have to do with the way I'm going for integration. So integration has only to do with the abundance of the species or useful pool space. It's not necessarily related to its colonization ability. The colonization ability comes in terms of it's a lottery model. So the more colonization ability the species has, the more species produces, so the higher likelihood that it will it's a patch-based model, so it will just produce more species. But if it lands on a patch that has a better competitor, it will lose to that competitor. But
Okay.
there will be more species that has more species.
yeah, okay. So it might have to do with the scale at which the local interactions may happening. It seems like it's an individual cell level where the species interactions are happening in your model. And if you have a more mixed local community where all of the individuals in a patch is interacting with one another, maybe you get a different kind of result. But it's difficult to give you any useful answer without knowing more about the specific model you have. So that's my way of dodging your question.
Okay, thank
But
you, Ted.
I like to talk more maybe at the